So this is how stored information on DNA becomes actions and proteins and life, human metabolism, everything that runs an organism. So the order of DNA bases, that unique order of DNA bases, which is 3 billion bases for a human genome, well, that determines the order of amino acids in a protein. The order of bases is read as triplet codons. So this would be 3 bases at a time. Each of these triplet codons codes for 1 amino acid. So that's the secret code of DNA. And it's not so secret because scientists have figured out these code words, these triplet codons that are equivalent to these specific amino acids. The genetic code of DNA is translated and becomes protein code. The amino acids are something we've seen before, monomers of proteins. So protein is a natural polymer made out of amino acid monomers. Every amino acid monomer has the same components, which are a central carbon that has an amine group, a hydrogen, a carboxylic group, and a side chain. Bonding together amino acids is called the primary structure. So that's the sequence of amino acids, and these are the monomers of protein. So the covalent bonds between amino acids, here's one covalent bond between the carboxylic acid of one amino acid to the amine group of another amino acid. This is a peptide bond. So every time there's a connection from the carboxylic acid that you can recognize the red oxygen to the amine group of the next amino acid, you can recognize by the blue nitrogen. That's a peptide bond right there. Putting these amino acids together as a polymer, that's called the primary structure. That's the linear sequence of amino acids. So what bonds amino acid monomers together are covalent bonds, and this is just two-dimensional. This is just put these together and you have a two-dimensional line. The primary structure of a protein is just the two-dimensional shape. That's just a line. So that's the amino acids bonded together. Well, this can fold into a three-dimensional shape, and part of the three-dimensional shape will include repeated motifs like an alpha helix that is a coil, like this. So this is a coil. We've seen this coil before. This is a spiral, or also a beta-pleated sheet. So a beta-pleated sheet is folding that looks like an accordion, and this will then curl around and have an overall three-dimensional shape that might include some alpha helix, and then this overall three-dimensional shape, though it looks like a big blob of a mess, proteins have a very specific three-dimensional shape that may include alpha helix, it may include beta-pleated sheet, and also includes a lot of random shape that is very specific, allowing for that protein to have a specific function, usually with all of this being a scaffold and then one section that's important, an active site, with specific functional groups in that active site that allow for a chemical reaction. The secondary structure is not the entire structure of the final protein. It's just a portion of the protein. It's a three-dimensional section of it. But one thing to notice is what maintains the structure of this alpha helix, what maintains this coil, are these dashed lines, those hydrogen bonds. The same is true of the beta-pleated sheet. What keeps this in the pleated three-dimensional structure are these hydrogen bonds. And so hydrogen bonds are important in maintaining the structure of proteins as well as the structure of the DNA double helix. It doesn't stop at the secondary structure though. That secondary structure is just a portion of the final three-dimensional structure. So tertiary structure is the final three-dimensional structure of a single polypeptide. So this is from just basically one long ribbon that is now folded into a specific three-dimensional shape. Even though this looks like a big tangled mess, this is actually very deliberate. And guess what maintains this structure of this big tangled shape? Hydrogen bonds mostly. There are a few covalent bonds in there that are between amino acid side chains that have sulfurs, but it's pretty much all held together by hydrogen bonding. This is one long polymer chain that is now folded in a specific three-dimensional shape, and what you have is amino acids that might have been very far apart in the primary structure, in the two-dimensional sequence. Well, when you fold things together, this puts amino acids closer together in the tertiary structure, whereas like this one here at the very end, if you follow this around and around and around and around, maybe 200 or 300 amino acids later would be this spot, and those become close together due to the three-dimensional folding. This is one enzyme structure, chymotrypsin. This is important in human digestion. So when you have quaternary structure, this is when you have more than one polypeptide. Myoglobin stores oxygen in your muscles, and this is one polypeptide chain, and it has a three-dimensional shape. And in the middle here is a heme group that contains iron, and then an oxygen molecule, a single oxygen is stored on that. In the middle, so this is equivalent to the active site, where you have all the rest of this three-dimensional structure is important, but really what the action is right in here, where the oxygen is stored. Hemoglobin also stores oxygen, but it's in your blood. So hemoglobin moves oxygen around from the lungs to the muscles and transfers oxygen to myoglobin. And hemoglobin is actually 4 of these. So having 4 of these myoglobins means we're talking about quaternary structure now. This single myoglobin is tertiary structure because it's only one polypeptide. It's three-dimensional shape. Now you have more than one polypeptide's three-dimensional shape. Now you're talking about quaternary structure when you have more than one sequence of amino acids, even though they're all 4 exactly the same.